Chromosome mutations are a new type of mutation that we're going to be talking about. Up until this point in the course, we've been talking about mutations at a single locus and the alleles that are present at a locus. Chromosome mutations are changes in chromosome structure, and there are three different categories that we're going to discuss: deletions, inversions, and reciprocal translocations. Deletions remove a section of a chromosome. So that is our wild-type chromosome structure, perhaps with some loci that we know about on it. A deletion is simply a breakage and rejoining of this within one chromosome that removes a segment. So the mutation event would produce a new structure, and there would be breakpoints that have broken and rejoined with an intervening piece lost. Inversions, on the other hand, again affect one chromosome, but when we have breakage and rejoining, we're not losing the intervening piece. We're simply having this piece with these two breakpoints and then have it inverted and rejoin. So if we were to draw this after the inversion event, we would still have A and C in their proper locations, but we would have B now, I draw it that way, with the two breakpoints. Because of the 5' 3' structure of DNA, when there's a breakage and rejoining within a chromosome, it actually has to flip over in order to line up 5' to 3' and rejoin. We normally won't draw them like this in the class. We'll simply draw them like this, showing where the breakpoints are. But in reality, this is what it would look like if we were drawing a map properly. Reciprocal translocations, on the other hand, are breakage and rejoining between two distinct chromosomes. So now I'll draw these non-homologous chromosomes as different colors. And a translocation event would be a break in one and a break in another, and then a swapping of the two, such that after the event, we would have this sort of structure. For example, where these two chromosomes have swapped tips. So this is a reciprocal translocation because there's a reciprocal exchange between the two chromosomes, breakage and rejoining, and we now have what used to be one chromosome now spread across two chromosomes, and same thing here. This black chromosome used to be one chromosome, but now that genetic material is now present on two chromosomes because of this breakage and rejoining event. In the next segment, what we'll do is we'll start looking at the implications for these different events once we start dealing with them in crosses and how they behave genetically. So we'll start by discussing deletion events. So as we mentioned, a deletion is simply a breakage and rejoining within one chromosome where there's a segment that's lost. Deletions are usually deletion heterozygotes. In other words, this event happens to one of the chromosomes, but in an organism that's a diploid, for example, you would have two copies of that chromosome and only one segment has been deleted in one of the chromosomes. If we took this organism, which we will call a deletion heterozygote, now heterozygote does not refer to alleles at a locus, but because we're saying it's a deletion heterozygote, we're saying that this organism is heterozygous for two chromosome structures. One homolog has a deletion and the other homolog does not. So this is a deletion heterozygote, and we will go on to talk about inversion and translocation heterozygotes in the same sense. Now heterozygous for chromosome structure differences, not alleles at a locus. Okay, so if we took this organism and say test crossed it to a little a, little b, little c organism with normal structures, normal chromosomes, what we would see in this cross is actually half of the progeny would have this chromosome structure, the deleted one, and then the other homolog coming from the other parent would have the little a, the little b, and the little c. These organisms, these offspring from this cross, would have the little b phenotype. And the reason for that is they don't have a big B allele present. The big B allele was in the deleted region of that chromosome, and as such there's only one allele that's present for the B locus. Now there's other things that are missing as well, but we don't know what they are because we don't have a variant locus in that region aside from the B locus. So this has not simply deleted one locus, it may have deleted many loci on either side of it, but we only recognize the one because we have the little b here that allows the little b phenotype to show up. Put another way, deletions act, in most cases, as recessive alleles of what has been deleted. So let's just write that down. Deletions act as recessive alleles of deleted loci. The other thing that we should think about now is what would happen, say, if we took this organism and allowed it to go through meiosis. Would there be an impact on recombination frequencies on this chromosome? And as we'll see in the next segment, there are impacts, and specifically we'll discuss how these chromosome structures pair at synapsis. So here's the same organism that we were just dealing with, this deletion heterozygote that also is heterozygous for a number of different loci here. This is what this organism would look like at synapsis prior to meiosis 1. These chromosomes have replicated and they have paired with one another based on their homologous sequences. The issue is, so if we draw synapsis as these little dots, the issue is that there's part of this chromosome which has no homologous sequence in the opposite deleted chromosome. So what we get at synapsis is a deletion loop, where the one chromosome, the normal chromosome structure, loops out, and the rest of the chromosome that does have a homologous pairing sequence on the deleted chromosome finds its partner in synapses. Because chromosomes are thin and flexible, they can maximize their pairing at synapsis. So we have this region here that is unpaired, and then we have regions that are paired, and we get a deletion loop as a result. Now, the question might be, what about recombination frequency? Will this have an impact on recombination frequency? And in fact it will. Because there is no synapsis in this region, crossovers are not possible in this region. However, crossovers are possible wherever synapsis is taking place. So if we looked at, say, the A to C recombination interval, that map distance, say we had a prior known map, say from a previous question where we had map distances that were associated with these intervals, the A to C interval in this case would appear to be less. And that would be because crossing over can happen here, or it could happen in this region here, single crossovers of course, to give us the recombination frequency between A and C. But we've lost a large amount of space, physical space, that used to be available for synapsis and crossing over. So as a result, we would see a decrease in recombination frequency between A and C. As a diagnostic, later on we're going to be discussing how we tell these different questions apart. A reduction in recombination frequency for known linked loci is one diagnostic that can point you towards a deletion. Now on the other hand, this will also apply, as we'll see in a moment, to inversion, so we need to know the difference between an inversion and a deletion, and we'll discuss that more when we talk about inversions. But one diagnostic for a deletion is that there is a reduction in recombination frequency between things that you know are linked on the same chromosome. The other key diagnostic here is inappropriate recessive phenotypes showing up. So in this case, that loss of that big B allele because of that deletion allowed the little b allele on the other homolog to express its phenotype. This is not a case of dominance per se, although this effect has a historical name called pseudodominance. Basically what's happening is this little b is now acting as if it were dominant because we were expecting a big B to be present here. But it's called pseudodominance or false dominance, Because what's really going on is we've lost this allele here, and as a result there's only one allele of the B locus that's present, and because there's only one allele, that allele determines the phenotype. So whether or not you recognize that it's called pseudodominance, or you just recognize and remember that deleted loci act as recessive alleles of what's been deleted, either way it doesn't matter as long as you understand the concepts. So ways to pick out a deletion: recombination frequency between known linked loci is decreased, and we have inappropriate recessive phenotypes showing up when we didn't expect them to show up.